In this lecture, we are going to look at factors that control ventilation, or factors that control our respiratory rate. So we've already looked at the mechanics of ventilation, how the changes in the diaphragm change the volume of the thoracic cavity, which changes the pressures and causes air to move. What we're going to look at now is what actually makes the diaphragm contract to begin with, and what are ways that we control our breathing rate, our respiratory rate. So we have to come back to this equation, and remember it can go either direction. For internal respiration, our CO2 is going to leave the cell and travel into the blood to form that bicarbonate ion and that hydrogen. And that bicarb, remember, leaves, and it's a very important buffer, so it'll buffer as it works its way back. And this hydrogen that's produced isn't any big deal, because remember that any hydrogens that are typically produced during internal respiration when CO2 loads, they're used to kick off the oxygen. Hemoglobin, remember, actually buffers all of those for us. So as long as we have, normal CO2 levels in our blood, we're getting rid of it, we're exhaling it when it gets to our lungs, we don't have a problem. But what if there's an issue? What if for some reason I'm unable to get rid of that CO2, so it starts to accumulate in my blood? Well, when it accumulates, it's going to change the pH. And again, we have the equation that we've looked at. So if you were to have like hypoventilation, slow, shallow breathing, what's happening is that CO2 you're not getting rid of it. So you're retaining CO2 and your pCO2 levels go up. Well, this is called hypercapnia, for too high CO2. And when that happens, all the CO2 basically only has one thing it can do. It combines with water and forms carbonic acid, and now it makes excess hydrogen ions, because now you have more hydrogen than you're trying to unload oxygen. And so this lowers the pH. Remember, if our hydrogen concentration goes up, our pH goes down, and we have what's called acidosis. Because it's your lungs that are doing it, we call it respiratory acidosis, which you'll probably see a lot. Now, if we were breathing really fast, like hyperventilating, then what happens is we're getting rid of CO2. So our CO2 levels are actually dropping, and you'll see shortly that the body tries to maintain CO2 levels. So if they fall, this equation actually shoots to the left, and you try to generate more CO2. Well, when that happens, now you're using up all these hydrogens. Well, if you don't have hydrogens, right, if your hydrogen ion concentration should decrease, then we know your pH goes up, and we would say you have alkalosis. So that could cause respiratory alkalosis, which you won't see as often as you will acidosis. And so this is a picture from your book. And again, hyperventilation, maybe a panic attack, certain medications. And so because you're getting rid of CO2, we need to make more. And so we're going to use up those hydrogens to make more, basically combine with the bicarb, so we can get CO2 back. If we have hypoventilation, on the other hand, too much CO2 is going to produce too many hydrogen ions, and then that is going to cause our pH to fall, which we would see with respiratory acidosis. So as far as what controls breathing, we mentioned already the diaphragm, right? The active phase is when it contracts, and we know that the phrenic nerve is the nerve that brings that information down to the diaphragm. Now, there's several respiratory centers, and depending on what book you read, you will see different names. In fact, this picture came from a different book. So to keep it simple, we are going to talk about just the inspiratory center, otherwise known as the DRG. Now, again, some books will say VRG, some will say DRG, and I have both in your notes, so don't get too hung up if it's the ventral group or the dorsal. There's two of them here, and nobody agrees. But we'll agree on the fact that we're going to call it the inspiratory center. And so that center has neurons that on their own depolarize. And when they depolarize, they are going to form that action potential or nerve impulse and send it to the diaphragm to cause the diaphragm to contract. So these neurons on their own spontaneously depolarize. So it's kind of crazy when you think about it. Like, remember the SA node generated its own action potential? So do these cells. And so we have this cyclic nature to it. And so typically when they depolarize, when they're on, what they're going to do is send information to the inspiratory muscles, primarily the diaphragm, and then the diaphragm contracts and inspiration occurs. And so we know that we typically are going to inhale for about roughly 2 seconds, and then we usually exhale for about 3. So they're on, the diaphragm contracts, and we inhale, and then they're off. And they're literally off. There's no activity. And so when you don't innervate the diaphragm, right, if you cut off the nerve impulse, the diaphragm relaxes and goes back to its dome shape. And that's why we exhale. And so they're on and they're off, and they're on and they're off. And so issues here are what can cause issues with like SIDS, and there's something called SUDEP, sudden death and epilepsy, where someone might spontaneously stop breathing. It's because they don't go back on. Anything that depresses your brain, this is in the medulla oblongata, right? So we're up here in the medulla oblongata. Well, anything that inhibits this area, like alcohol overdose or barbiturates or sleep aids or morphine, will actually suppress the system, and then it never turns on, and then you stop breathing. So also know that there's accessory muscles that are stimulated as well. So primarily those intercostal muscles, the external and internal, and just know that that can stimulate those anytime we need to take deeper breaths. So we're back to chemoreceptor reflexes. They're very important in controlling respiratory rate. We looked at them already for blood pressure. And when we look at the chemoreceptors, there's a few differences between these and what you previously learned. For one thing, the respiratory system has central chemoreceptors and peripheral. Now, the peripheral are your usual suspects. These are the ones that are out here in the carotid body, remember, carotid sinus, and in the aortic arch. So you kind of learned these. But the central ones are right up in the medulla, and the central ones are right around that respiratory group, that inspiratory center. So they will directly affect the inspiratory center. When we look at chemoreceptors, we're still looking at CO2, O2, and hydrogen. But a second difference is that your breathing rate, your respiratory rate, can change based on whether your CO2 levels are high or low. So previously, when we looked at blood pressure, we said it was only, chemoreceptors were only stimulated if your CO2 went up, your O2 went down, or your hydrogen ions went up, right? Your pH went down. Well, now we know that high or low levels of these can change our breathing rate. However, you're going to see that we are most sensitive to CO2. So you should star these central chemoreceptors because they are the most important ones. And the central chemoreceptors are only going to respond to CO2. So a lot of times you might think that the reason we breathe is because we need to take in oxygen. This is called our respiratory drive, like what makes you breathe in. But in fact, it's not that at all. It's because we have to get rid of CO2. So we are most sensitive to CO2 levels. Now, when we look at these peripheral ones, they mainly respond to your blood oxygen levels and your blood pH. And then they send information up to the medulla oblongata, just like we already learned. So remember, if you look at the carotid bodies, they're sending their information up through cranial nerve 9, right, glossopharyngeal, and the aortic arch is sending it up through the vagus nerve. So they still use the same nerves, by the way, for those peripheral. So CO2 is the most important factor for breathing, and we're going to change this to say 35 to 45. We're going to keep that simple. So 35 to 45 mmHg is where your blood, arterial blood CO2 should be. So when we talk about this, we're always talking about arterial blood. So if you hear ABGs, that means arterial blood gases. So usually they go after it for a radial artery or brachial artery because they need the arterial blood, not the venous. Now, we already said that when our CO2 goes up, our pH goes down. That was this equation we looked at. Well, what happens is that the CO2 is actually able to go through the blood-brain barrier. So remember, you had that blood-brain barrier. It was a layer of astrocytes that protected your brain, and not a lot of stuff gets through. Well, CO2 does. And so what happens is when it goes through the blood-brain barrier, it does this reaction. It combines with water and forms carbonic acid, and it makes hydrogen ions inside your cerebral spinal fluid. Now, cerebral spinal fluid does not have buffers. It has very little buffers. So just tiny changes in CO2 can really change the hydrogen ion concentration and change the pH pretty quickly. And it directly stimulates those central chemoreceptors around your medulla. So the reason CO2 is critical is because it can pass through the blood-brain barrier. And then again, it changes the pH of your CSF. And when you come in and you change the pH around your cerebral spinal fluid, that directly stimulates the medulla, the inspiratory center. And then it's like, oh boy, we need to get rid of CO2. So you increase your rate and depth of breathing. You probably have a higher tidal volume and you increase your respiratory rate because right, you're trying to get rid of all that excess CO2. You had too much. So now you're going to breathe faster to try to eliminate it, to basically get it out of your body. So remember, hypercapnia is when our CO2 levels are too high. And so we know then that would cause an increase in my respiratory rate as well as my tidal volume. And again, we are most sensitive to CO2. In fact, a CO2 going up by 5 mmHg would double your respiratory rate to get rid of and flush it out. Now, if you see hypocapnia on the other hand is low CO2. So when we're looking at CO2, we need to maintain homeostasis. Anytime maybe we're not able to eliminate the CO2, or maybe there's an issue with CO2 rising, like you went underwater and were swimming underwater or holding your breath, but the CO2 levels start to increase, we know that will then go through the blood-brain barrier. It will stimulate the CSF and cause an increased respiratory rate to get rid of it, to flush it out. If my CO2 levels fall, usually I'm like breathing too fast often, then what's going to happen is we need to maintain the CO2 because we have a range, right? 35 to 45. So now we need to bring the CO2 back up. So you would actually slow down your breathing. You could even stop breathing momentarily. So a lot of times for swimming, before you take off in competitive swimming off the starting block, you might do a real quick hyperventilation. So, and when you do that, you're lowering your CO2 levels. Because if you drop your CO2 down to 35-ish, you won't feel the need to breathe again until it's around 45. So what does that mean? That means you can stay underwater longer. And if you're swimming, it's better to stay underwater longer than to come up too soon. Well, the problem with that is that if you hyperventilate and you lower your CO2 levels, one thing, you're going to have to bring them back up. Well, anytime my CO2 falls, it causes this vasoconstriction of blood vessels, of my cerebral blood vessels. I can't talk today. Well, the last thing you want to do is vasoconstrict blood flow to your brain. You're probably going to pass out. And so if anyone is hyperventilating, a lot of times they get lightheaded and feel like they're going to pass out because of that. And probably the last place you want to pass out is underwater. Now, PO2 does not affect the central. It only affects the peripheral. And it doesn't do much. In fact, usually if your PO2 is falling, all it does is make your central chemoreceptors a little more sensitive to CO2. In fact, your PO2 levels in your blood have to drop to about 50. So think about that. Normally, right, your PO2 is 104. It has to drop to half of that before you would feel like you needed to breathe. Because again, we breathe anytime our CO2 levels go up. That's the stimulus for breathing. Oxygen, the critical point, is around 50. So typically a PO2 has to fall to 50 before it becomes the stimulus for breathing. The one population this is not true for is people with COPD, chronic obstructive pulmonary disease. They always have high CO2. And because their CO2 is elevated all the time, their central chemoreceptors don't really pay attention. They kind of start to ignore it. And in those individuals, oxygen is their respiratory drive. But remember, you're not really going to feel the urge to breathe until your PO2 falls. So these individuals are usually on a low flow oxygen, because if you crank it up too high, they won't breathe for a little while because they won't breathe until they hit around 50. That's like the critical point. And we also know that blood pH is not significant for respiratory rate. And that's because the hydrogen ions don't get through the blood-brain barrier. So it can stimulate the peripheral chemoreceptors we mentioned. But again, the peripheral ones aren't the main players when we're looking at changes to respiratory rate. So again, anytime we're looking at our respiratory rate, and if my CO2 levels go up, that's going to stimulate an increased respiratory rate to get rid of it. And if I have that hypocapnia, right, so hypo is too low, now the issue is they're falling, so I'm going to slow down or even stop breathing, and my cerebral vessels often vasoconstrict. So our respiratory rate is primarily determined by our CO2 levels. Now, there's some other reflexes that affect it. So again, when we're looking at what makes us breathe, our respiratory drive, the number one factor is our CO2 levels because it affects the central chemoreceptors. My O2 and hydrogen out in my blood affect the peripheral, but they're not main players in controlling respiratory rate. And there's some other things that change our respiratory rate. So the first one is like irritant receptors. So irritant receptors are things that respond to like pathogens or dust. These are things that make you cough or make you sneeze. So our breathing is different when we're coughing or we're sneezing. The other thing you see here is a stretch receptor. It's called the Hering-Breuer reflex. This is a stretch reflex. If you take a deep breath in, you'll reach a point that you stop inhaling. You just stop. And then your lungs naturally recoil and you exhale. So this is done to prevent your lungs from basically overinflating. And then the higher brain centers like our hypothalamus and cerebral cortex, they also affect our breathing. Hypothalamus more with emotions. So we might change our breathing when we're very happy or excited or sad. So that could be tied in with the hypothalamus. And then the cerebral cortex is voluntary control. So, when I'm talking and giving a lecture, I don't take a breath normally, as you might notice. Or maybe I'm playing the trumpet or I'm singing. So I can voluntarily change my breathing rate and alter it to do voluntarily change it momentarily. So that wraps up our lecture on factors that control pulmonary ventilation.